Hello. Today we continue talking about the fertilization, okay, and we'll understand more details about the fertilization. We need to understand that straight after the fertilization, there is the machinery of the protein synthesis start to work in high intense, and try to use the messenger RNA, which already passed by the oocyte and exists in the oocyte cytoplasm. So the protein synthesis in the embryos of the sea, for example, the sea ocean, in experiment, they tried to check the protein synthesis in the presence and absence of an important inhibitor for transcription, which is the actinomycin. So it's experiments that show that when you can see the graph, so actually it is on the x-axis, you can see the hours, it's a time after fertilization. And then you see on the y-axis, the valine incorporation in the protein. So that's a reflection. The valine is one of the amino acids. That's a reflection of the protein synthesis. So if the valine is incorporated inside the protein, and it can be measured radioactively by using carbon-14. So if it's incorporated in the protein, that means the process of protein synthesis is working, okay? So it's like an indicator for the process of protein synthesis. So you can see that without the use of actinomycin, without the use, the red graph, you can see the red line chart. So you can see without the use of the actinomycin, you can see that a protein synthesis increases over time after fertilization, increases over time, reaching a peak around 25 hours after fertilization. While after the use of the actinomycin, you can see, since it's an inhibitor of transcription, you can see that the process of transcription is markedly diminished or decreased. So this graph shows clearly that there is a burst in the protein synthesis after fertilization. So the fusion of the genetic material leads to actually occur after the sperm enters the egg cytoplasm. And basically, what enters from the sperm is the head part with the part of the middle piece, which contains the centriole and actually the proximal centriole. So actually, the male pronucleus separates from the tail, and it needs to rotate 180 degrees so that the sperm centriole, which is actually the proximal centriole that is in the neck of the sperm, will be located in between the sperm egg and the sperm pronuclei and the egg pronuclei inside the oocyte. So now the sperm centriole, since it is located in the middle between the two pronuclei, it will act as a microtubule organizing center in order to make the spindle fibers to get the zygote ready for the process of division. So the microtubule extends throughout the egg and contact the female pronucleus. And the two pronucleus start to migrate toward each other until forming until fused and forming the diploid nuclei. And DNA start can begin either in the pronuclear stage during migration or after the formation of the zygote nucleus. And this actually depends on the level of the calcium released early in the fertilization. So let's talk about the internal fertilization in mammal, and we need to understand the difference between translocation and capacitation. You need to understand that in the internal fertilization of the mammal, why it's internal? Because the fertilization occurs internally inside the female. So the female actual reproductive tract is not a passive conduit. It's not a tube that only help in the transport in which the sperm moves, like the oviduct. But this tube in which the sperm race until reaching the ovum is a highly specialized set of tissue that actively regulate and transport and even lead to maturation of both gametes. So what is a translocation? Actually, translocation, it is the translocation of a sperm from the vagina to the oviduct, and this involves the processes that work at different time and places. So the uterine muscle contraction are critical in getting sperm into the oviduct. And of course, as we previously understood that the sperm motility is as we understood before, one of the major factors, but actually sperm motility, due to its flagellar action, actually it's a minor factor in getting the sperm into the oviduct. So the major factor is the uterine muscle contractions. Actually, the motility is required for the sperm, for example, the mouse sperm, to travel through the cervical mucus and for sperm actually to encounter the egg once they are in the oviduct. So what about capacitation? So actually capacitation is a set of physiological changes that allow the sperm to be competent to fertilize the egg. So some modification need to be done for the sperm to be competent and to be able to fertilize the egg. Although some sperm can reach the oviduct within a half hour after intercourse, speedy sperm doesn't mean that it can fertilize the egg, and actually may have little chance of fertilization since they are not undergo capacitation. So we understand now that capacitation is important for the ability of the sperm to fertilize the egg. Ibrahim, could you mute your mic, please? Okay, so, and this proves that speedy sperm, even speedy sperm, and can reach the ovum very fast, even if they could reach the egg very fast, they might be not able to fertilize the egg because they didn't undergo capacitation. So as we said, capacitation is a set of physiological change, as we'll discuss now. It will help the sperm to be competent to fertilize the egg. So in capacitation, you have 5 sets of molecular changes that need to take place for the sperm in order to be competent. The first thing, there are some alteration of the cell membrane, the sperm cell membrane, by the removal of cholesterol by the albumin protein in the female reproductive tract. So the female reproductive tract, which is the oviduct, for example, have albumin protein. This albumin protein will remove the cholesterol in the cell membrane. Of course, why? Because the cholesterol in the cell membrane of the sperm could interfere with the process of attachment of the sperm on the cell surface of the ovum. So the removal of the cholesterol will disrupt the lipid arrangement of the sperm cell membrane. And this will leave lipid raft cluster over the anterior sperm head. These lipids contain protein that actually will help the sperm to bind to the zona pellucida and participate in acrosome reaction. So in other words, if the cholesterol is not removed from the anterior part of the cell membrane of the sperm, this will interfere with the binding of the sperm to the zona pellucida, and hence there will be no acrosome reaction. And this is the first molecular event occur for the sperm. The second molecular event, some proteins or carbohydrates on the cell surface will that block recognition sites for sperm protein that bind to zona pellucida are lost. So the sperm, this molecular change will enable the sperm to recognize the zona pellucida. In other words, in case there are protein on the surface of the cell membrane of the sperm that could not recognize the zona pellucida, this molecular change will ensure that these particular proteins or carbohydrates are lost before contact with the ovum. Any question يا جماعه لغايه دلوقتي؟ اي سؤال على الجزء بتاع الكاباسيتيشن او الترانسلوكيشن؟ ما فيش اسئله؟ Okay. The third molecular change, which one of the important as well molecular change, the membrane potential of the sperm membrane changes. It becomes more negative. And that's why, because the calcium ion leave the sperm while the calcium channels are open as well. So you have like the ions that change its locations. So calcium, basically calcium ions and bicarbonate ions are critical in activating the cyclic AMP production and facilitate the future membrane fusion event of the acrosome with the zona pellucida. The fourth molecular event is a protein phosphorylation occur. So you have two kinds of protein, which is called heat shock protein or chaperones, two chaperone proteins that need to migrate to the surface of the sperm head when they are phosphorylated. So this phosphorylation is important for triggering these two chaperone protein to migrate to the surface of the cell membrane of the sperm. Why these proteins are important? Because they play an essential role in forming the receptor that bind to the zona pellucida. So in case that there is no phosphorylation for these two chaperones, the sperm will not be able to bind to zona pellucida. The fifth molecular change that occurred to the plasma cell membrane, actually the outer acrosomal membrane changes and come in contact with the cell membrane in a way that prepare it for fusion. وعشان نفهم الجزء ده يعني احنا عندنا كان بيبقى برضه نشوف بالقلم كده لو دي النيوكليس بتاعت السبيرم، وده طبعا دي بيبقى عندها هنا في النك البروكسيمال سنتريول وعندها الديستال سنتريول اللي طالع منه الفلاجيلا. بعد كده عندنا هنا الميتوكوندريا لو انتم فاكرين، وبعدين كل ده، وبعدين عندنا هنا كده ده الاكروسوم. فعندنا السيل ممبرين بيبقى عامل كده اهو عليهم، لو انتم فاكرين ده الاستراكشر بتاع السبيرم، ده السيل ممبرين ماشي كده لغايه تحت. فالموليكولر ايفنت رقم 5 بمنتهى البساطه ايه هو؟ ان السيل ممبرين اللي موجود هنا اهو، السيل ممبرين لو كبرناه الجزء ده هيبانوا زي خطين. الاوتر هو السيل ممبرين بتاع السبيرم، الانر هو السيل ممبرين بتاع الاكروسوم. بمنتهى البساطه اللي بيحصل ان الاوتر اكروسومال ممبرين، لان ده يعتبر الاوتر اكروسوم خلاص، بدل ما هم بيبقوا بعاد عن بعض بيقربوا خالص من بعض بهذه الطريقه، بيقربوا خالص. عشان كده بيقول لك ان الاوتر اكروسومال ممبرين comes in contact with the sperm cell membrane in a way prepared to the fusion. فهمتوها دي يا شباب ولا لا؟
Yes, دكتور مش الحال.
Okay. So these actually the 5 events that are crucial and collectively called capacitation. So it is a physiological change, basically molecular changes that enable the sperm to be competent in order to fuse with the egg. So if we look closely to the model of capacitation and a part of the molecular event that occur inside the cell membrane, inside the sperm, you can see that we have albumin in the oviduct that pull out the cholesterol. There is a cholesterol efflux outside by the help of the albumin which occur in the oviduct. This is one of the events. This actually will promote the opening of the bicarbonate ions to be internalized, as well the potential difference will change due to the efflux of the potassium ion to the outside, and as well the internalization of the calcium ion. So this the internalization of calcium ion together with the internalization of calcium ion together with bicarbonate ions will activate the adenylate cyclase enzyme which exists in the cell membrane that convert adenosine monophosphate AMP into cyclic AMP. The increase of the concentration of cyclic AMP in the cytoplasm will activate a kinase which is called pKa, and this pKa is important in activating another kinase which is called protein tyrosine kinase, and they inhibit actually the counterpart of this which lead to its dephosphorylation, which is a phosphotyrosine phosphatase. In order to sustain the phosphorylation signal, it needs to inhibit the phosphatase. Both these events will activate protein tyrosine phosphorylation on the protein, leading to the process of capacitation. And as we said, the change in the membrane potential due to the efflux of the calcium will lead to a hyperpolarization of the membrane, which add in the process of capacitation. فاهمين يا شباب الموليكولر بعض من الموليكولر ايفنتس اللي بتحصل جوه السبرم ولا لا؟
Yes.
Okay. So the coda or the conclusion of that part, actually fertilization is not a moment or an event. يعني هو ليست لحظة بتحصل حاجة الاثنين بيتقابلوا وبيحصل fertilization زي ما احنا كنا عارفين من زمان. But it is a process of carefully orchestrated and coordinated events, including contact, fusion of gametes, fusion of nuclei, activation of development. And if we want to dissect, there is capacitation, there is translocation. So there is a lot of molecular event. It is not just a contact between gametes. So it is a process whereby two cells, at each of them at the verge of death. يعني السبرم والاوفا لو حصلش fertilization هيموتوا الاثنين في وقت قليل. But once they can unite, they create a new organism with numerous that will soon develop into an organism with numerous cell types and differentiate into organs. So it is just the beginning of serious cell-to-cell interaction that characterize the animal development. Now we'll talk about some of the early invertebrate development and some of the cell specification. So I'm reminding you of the early developmental process we have discussed in the first lecture. And as well, we now, after fertilization and the formation of the zygote start to increase in the number of cells by a process of cell division. And actually the cell division or cell dividing in that stage after the fertilization have a peculiar name, which is called cleavage. So fertilization, and we understand in the few slides why it's called, how it's different than the normal cell division. So actually cleavage follows fertilization, and it is a set of mitotic division, the typical mitotic division, in which the volume of the zygote, you have a volume of the zygote, will be divided into numerous smaller cells. And these smaller cells, now we have to say it have a specific name which is called blastomeres. وده يمكن أول فرق هنلاحظه ما بين السيل ديفيجن العادي والكليفج. في السيل ديفيجن العادي اللي بيحصل ايه؟ أنا بيبقى عندي cell, one single cell, and then undergo a mitotic division. It will give rise to two cells بالمنظر ده صغيرين, and the two cells دول will grow until reaching the same size of the parent cells. Okay, this is the normal cell division. لكن في الكليفج الوضع مختلف. الوضع انا عندي زيجوت اللي هو السيل بهذا السايز, وهذا الزيجوت بيبدا يتقسم لخليتين, two cell stage. الفكره هنا ان التو cell stage will not grow عشان يوصلوا لحجم الزيجوت. لا, ده دول هيخشوا على طول successive cell division ويصغروا اصغر, وهكذا يصغروا اصغر. يبقى بالتالي في الكليفج there is a leak in the growth phase of the cell cycle in the cell cycle. وده اللي يفسر لنا ان الفوليوم بتاع الزيجوت actually الفوليوم have been divided بالمنظر دوت. يبقى الفوليوم have been divided into smaller volume in each smaller cells, which is now called blastomeres. So the zygote genome now, it needs to be dormant. It's not active. The zygotic genome doesn't function in early cleavage because the zygote is only concerned at that stage of the early cleavage stage to increase in number, not in expression, in gene expression. So the rest of the zygote is divided into half, then quarter, then eighths, and so forth. The division is accomplished by abolishing the grass period, as I have just said, between cell division. So actually, there are no G1 or no G2 phases of the cell cycle. And this explains why these cells or the plastomere tend to go smaller in size, okay? So the cleavage, actually, the cleavage of the nuclei occur at a rapid rate, which will never seen again in the course of the development of the embryo. فاهمين الجزء ده يا شباب ولا لا؟
Yes.
Okay. So let's see here. You can see here that a frog zygote can divide into 37,000 cells in just 43 hours, while a Drosophila zygote can divide to 50,000 cells in just 12 hours. And you can see here in this graph, which have the x-axis have hours at 15 degrees, which is the optimum temperature for the development of this embryo. And you have the number of, here on the y-axis, you have the number of cells per embryo. So you can see here, starting the first point here shows here, it is a point of fertilization. You can see that the number of cells is dramatically increased during the cleavage period, because there is fast rate of cell division, fast rate and increase in number. While the following process, which is called the gastrulation, the rate of division is a lot smaller, because the fast rate is actually here at the stage of cleavage, which soon this rate is decreased a lot, although there is the cell division, but it is decreased in the stage of gastrulation. And this actually shows the early development of the frog rana penis. So let's talk closely now about from fertilization to cleavage. Of course, with the rapid cell division, the ratio of the cytoplasm to nuclear volume gets increasingly smaller. وعشان نفهم الجزء ده يا شباب، تعالوا نرسم الرسمه دي. انا عندي الزيجوت كان عامل كده. ادي الزيجوت. النيوكليس بتاعته كده. حد يقدر دي النيوكليس. حد يقدر يقول لي الريشيو سايتوبلازم تو نيوكلير بتطلع قد ايه تقريبا؟ تقريبا الريشيو كام لكام؟ حد يقدر يحسبها من الرسمه دي؟ قد ايه سيتوبلازم قد ايه نيوكليس؟ 1 to كام مثلا؟
1 to 6 يمكن.
يعني انت دقيق زياده عن اللزوم، خلينا نقول تقريبا يعني ان السيتوبلازم مثلا هنعتبره 2 او 3 to 1. صح كده ولا لا؟ السيتوبلازم تو نيوكليس، صح كده؟ تقريبا يعني، اوكي؟
يس.
طيب مع فيرست كليفج هنلاقي الموقف ايه؟ النيوكليس الحجم بتاعها مش هيتغير. خليتين مثلا والنيوكليس تقريبا عامله كده. الريشيو تقريبا اختلف ولا لا؟ السيتوبلازم تو نيوكلير ريشيو اكيد وده يفصل لنا ان الريشيو during the rapid cell division, because there is no G1, there is no G2 in the cell cycle, the ratio of cytoplasm to nuclear volume get increasingly smaller as cleavage progresses. So this decrease in cytoplasm to nuclear ratio is crucial in the activation of certain genes. يبقى العلماء لقوا ان لان احنا اتفقنا early in cleavage there is no gene, there is no expression of genes. But when you decrease the ratio from cytoplasm to nuclear volume, this will activate a set of genes later in cleavage. For example, they found that in the frog species of the frog, one species of frog which is called Dinopus levius, the transcription of a new genes is not activated until the 12th division, the division, the cleavage number 12, which around the mid-blastula transition stage. وده لأول مرة بيظهر معانا مصطلح اسمه blastula. What is blastula? Blastula is meant to be the end stage of cleavage. So when reaching mid-blastula, we are around the halfway to the end stage of the cleavage. They found that after 12 division, the activation of genes doesn't start until the cleavage number 12. So thus cleavage begin soon, actually, as we all know by now, the cleavage begins soon after fertilization and ends shortly after the mid-blastula stage, when the embryo achieve a balance between the nucleus and cytoplasm. And of course, the balance between the nucleus and cytoplasm, it's meant here the balance between the ratio of the nucleus to cytoplasm. So the transition from the fertilization to the cleavage actually is caused by the activation of a set of factors which is called mitosis promoting factor, which actually are proteins. So this mitosis promoting factor is responsible for restarting the meiotic cell division in the mitotic cell division in frog embryo or frog egg. So the generated blastomeres due to the cleavage will generally progress through the cell cycle very fast, and only will have two stages of the cell cycle: the mitosis, the M phase, and the S phase, which is the B synthesis. That's why the cell cycle of these blastomere is called biphasic cell cycle, because it doesn't contain the normal 4 stages of the cell cycle. So this MBF activity at the early blastomeres is high during the M and undetectable during the S. And this very clearly gives the clue that the cells undergo a vigorous mitotic division. And you can see clearly the difference here. You can see here, this is actually the normal cell cycle. You have M, you have G1, you have G2, you have S. Okay, the only difference here, the major difference between the normal cell cycle and the cell cycle which actually occurring during cleavage, you can see that you have active CDC protein kinase, which actually are part of the MBF, and these active CDs are very active, constitutively active. So as you can see here, the cyclin B here, it is degraded at some stage. Once it degraded, this will give a clue for the cell to go for interphase and restart the cell cycle again. So it will skip the G1 and G2. So the cyclins, these cyclins allow the progression to the M phase, while cyclin, the degradation due to its degradation, allow it to straight away to pass into S and skipping the G1. Okay. So cells that are differentiating are usually taken out of the cell cycle, or in normal cases, cells stop cycling. Why stop cycling? Because at some point after division and cycling, it can differentiate, and once it differentiates, it go into a quiescent stage, which is called G node or G0. يعني لو خدنا مثلا during development، انتوا عارفين يا جماعه ان عدد النيرف سيلز في جسمنا هو عدد ثابت لا ينقسم، فبالتالي during development بيتكون النيرف سيلز دي، وبعد ما يتكون العدد الثابت لكل اورجانيزم، هذه الخلايا بتخش في مرحله الجي نود او الجي 0 to stop further cell division. فاهمين على السلايد دي ولا لا يا شباب؟ ولا في حد عنده سؤال في السلايد دي؟
دكتور بس كيف قلنا بيصير في عندنا ديجراديشن للسايكلين؟
اوكي، وجود السايكلين معناها ايه يا ابراهيم؟ وجود السايكلين معناها ايه؟ وجود
السايكلين انه السيل تروح على الميتوسيس، على الميتوتيك فيز.
لا وجود السايكلين بمنتهى البساطه، بص تعالى هنا، الام هنا مش في سايكلين هنا ولا لا؟ وجود السايكلين عموما خلاص بيساعد الخليه ان هي تبدا تعمل سايكلينج انتو ذا 4 نورمال ستيجز. فهمت الفكره ولا لا؟ وجودها. لكن وانس حصل ديجراديشن هنا للسايكلينز كلها، وخلي بالك هو ما حددش انه سايكلين، ديجراديشن للسايكلين عموما، فبالتالي ما عنديش سايكلين دي اللي يدخل الخليه في جي 1، فبالتالي الخليه بتروح اسكيب الجي 1 وتبدا تخش تاني اكنها بتعيد تاني من الاول الانترفيز. فهمت الفكره؟ وتبدا ايه الام فيز تاني. فهمت الفكره؟
اوكي.
في حد عنده سؤال تاني؟ So actually the mitosis promoting factor it contains two subunits, as we can see from the figure here. It's a cyclin B and CDC2. So a large subunit, which is the cyclin B, and the small subunit, which is CDC, which is cyclin-dependent kinase. Cyclin B actually is encoded by messenger RNA that came from the oocyte. So it was not transcribed. It is already exists in the cytoplasm of the oocyte. And this will enable this cyclin B to enter the cell to enter mitosis. While the kinase will activate mitosis by phosphorylating other target proteins. Hence, this phosphorylation hence will lead to, and even because actually the chromatin is condensed, the nuclear envelope will be depolymerized, and the organization of the mitotic spindle. Therefore, the cell cycle is actually independent of the nuclear genome, because actually, why it's independent of the nuclear genome? Because all the components of the protein that initiate the mitosis in the zygote came actually from messenger RNA in the cytoplasm, which was exist before the formation of the zygote, because they came from the ovum. So that's why the cell cycle at that stage is independent of the nuclear genome. However, the as the process of cleavage is going on and on and on, the amount of the messenger RNA which is exist in the cytoplasm that came from the ovum, or collectively, which is called cytoplasmic components, are used up. So that's why we have said early today that the expression of the gene at the early stages of the cleavage is not occurring. But here, as the cytoplasm component is being used, so the nucleus begins to synthesize them again. So the egg cytoplasm actually will be, due to the division of these cells, will be divided into smaller and smaller and smaller cells, which now is called blastomeres. But the total volume of organism is remain unchanged. So as we said, cleavage goes on during the cleavage process, aiming to form a stage, the final stage of cleavage, which is called the blastula stage. But we have a mid-blastula transition stage, and this represents the point at which, after that point, the genes start to express genes, because the cytoplasmic component have been consumed. So let's collect the events that occur during the mid-blastula transition stage, because it is almost at the mid of the cleavage process. So we have the first event, which is of course, the ratio of cytoplasmic to nuclear volume get increasingly smaller as cleavage progresses. The second, the G1, after the mid-blastula stage, the G1 and G2 are added to the cell cycle. And this will permit the cells to grow. And actually, after the 12 cleavage in the xenopus, for example, and after the 14, for example, in Drosophila, it differs. The G2 will be added after the 14th cleavage, while the G1 will be added after the 17th cleavage. The third event, actually, the synchronization of the cell division is lost. So not all the cells will divide at the same time, because before the mid-blastula stage, the cell actually synchronized and all divide at the same time. 2 cells, 4 cells, 8 cells, and so on, until reaching the mid-blastula transition state. After that, the synchronization is lost, and each group of cells will divide in a different time. The nuclear genes, as we said before, the nuclear genes begin to be transcribed after the mid-blastula stage, or even starting by the mid-blastula stage, transition stage. And of course, due to the fact that nuclear genes will start to be transcribed, of course, new messenger RNA are needed or transcribed, because they have to be ready for the next process, next event or next stage, which is the gastrulation. And one of the important events that occur, that blastomeres need to be organized and take a specific position in the embryo. So that's why these plastomeres need to be motile. They just move around. So what about the cytoskeletal? What is occurring for the cytoskeletal mechanism during mitosis? So generally, the mitosis, as you already know, is a function of two coordinated processes: a karyokinesis, which is the mitotic division of the nucleus, and the cytokinesis, which is the division of the cells. And you can see you have two perpendicular rings that help in performing these two processes. You have two mechanical agents: a mitotic spindle, which is comprised of the tubulin protein, and you have a contractile ring, which is formed of the actin protein. So we have a contractile ring. You can see this is the contractile ring, which helps in the division of the cytoplasm, and you have a tubulin that which is the mitotic spindles, which is transversing the contractile ring, and that's why they appear perpendicular to each other. And these two mechanical agents will aid in the process of the mitosis. So as we said, the mitotic spindle and the contractile ring are perpendicular to each other. And this contractile ring will generate the furrow, the cleavage furrow, which will soon bisect the blastomere into two smaller genetically equivalent blastomeres. You can see that the difference, this small table describing the difference between the karyokinesis and cytokinesis. Of course, we have said the mechanical agent, and we have said the major protein composition of these two agents. The location, actually, because you can see that the cytokinesis with the contractile ring actually occurring in the cortical cytoplasm, while the microtubules occur located in the central cytoplasm. One of the and we need to understand some of the drugs that have been used for inhibiting this process. Two major drugs like colchicine and cytoskeletal B are used to inhibit either the karyokinesis or the cytokinesis. وده يخليني اسالكم سؤال يا شباب. ايه اهميه ان احنا نبقى عارفين ان في drug can inhibit either the mitotic spindle or inhibit the contractile ring. وليه العلماء فكروا ان to develop a disruptive drug that can inhibit either the mitotic spindle or either the action of the contractile ring. حد عنده فكره ليه العلماء فكروا في الدراجز دي؟
هلا ليش بيعملوا انهبيشن مش كرمال
لا انا ما بقولش
او شيء؟
ايوه انا مش بسال السؤال ليش بيعملوا انهبيشن، انا بسال السؤال ليه العلماء فكروا ان هم يطوروا دراج يمنع الكاريوكاينيز او يمنع السيتوكاينيز. انت كنت بتقول حاجه فيها كانسر كده.
يس.
ايوه
منشان يقدروا يوقفوا الكانسر او شيء.
ممتاز، لان برضو الميكانيزم بتاعت الكانسر هتلاقيها قريبه جدا من الميكانيزم بتاعت الكليفج، صح كده ولا لا؟ لان دي خلايا بتنقسم سريع جدا، فهم راحوا مطورين عشان يقدروا تو اكسبيرمنت الدراجز بتاعتهم. فمنتهى البساطه ممكن يجيبوا فروج امبريو وعنده كليفج بكميه كتيره جدا، فبالتالي ايه اللي هيحصل؟ لما يحصل كليفج بكميه كتيره جدا يجربوها في المعمل، فيقدروا يجربوا اذا كان هذا الدراج هيقدر يمنع الفورميشن اوف مايتوتيك سبندل او كونتراكتايل رينج قبل ما يجربوا هذا الدراج على البني ادمين. فهمتوا يا شباب ولا لا؟
Yes.
It's interesting، صح كده؟
Yes.
تمام. So we understood now the importance of developing a drug that could be experimented on experimental models like frog, for example, and the development of the frog to develop drug that can inhibit the cell division. فهمتوا النقطة الأخرانية دي يا شباب؟
Yes.
Okay. So let's talk now about the patterns of embryonic cleavage. We understood now the cleavage as a definition and why it's important for decreasing the cytoplasmic to nuclear ratio, and in order to this decrease in the ratio will promote the expression of genes, especially at the mid plastular transitional state. But we need to understand pattern of cell cleavage because it differs based on the organism. So basically, different organisms, different group of organisms undergo cleavage in different ways. And there are two main factors that, two major parameters that determine the pattern of cleavage of the organism or the species. Actually, the amount and distribution of the yolk protein, and actually yolk, which exists inside the cytoplasm, actually it is the nutritive material which give the energy needed during the process of development of the early process of development of the embryo. So the amount and distribution of the yolk, one of the major parameters that control the pattern of cleavage, as well as the factors in the egg cytoplasm, which of course include the messenger RNA, which exist, the transcription factors that influence the angle of mitotic spindle. So understood now that the mitotic spindle, the angle at which it's located inside the cytoplasm will determine, actually the angle and the timing will determine the pattern of the cleavage. So the amount, as we said, the amount and distribution of yolk will determine where the cleavage can occur and the relative size of the blastomeres, okay, as we'll see later. So usually the egg is divided into two regions. A region, usually, generally, a region that is yolk-free, okay, in which the division will go through that region very fast, at faster rate than the opposite region, which majorly will contain more yolk material. Okay, so we have a yolk-rich pole or a yolk-rich half, which is called the vegetal pole or the vegetal hemisphere, and we have a yolk-less pole or a yolk-less hemisphere, which is called the animal pole. So the zygote nucleus usually is displaced in the animal pole, while the and of course in general the yolk inhibit cleavage. فاهمين يا جماعه هنا التو كرايتيريا اللي على اساسها الباترن اوف كليفج بيتحدد ولا لا؟
Yes.
Yes.
So let's now, since the amount and distribution of yolk is very important in determining the pattern of the embryonic cleavage, we need to understand the different types of egg. So usually the eggs of sea urchin and mammalia, ourselves and snails, have equally spaced spurs, very few amount of yolk, okay? And this type of egg actually is called isoleucithal egg. And basically, leucisthal means yolk. Iso means equal. So that's why it's equal distributed distribution of yolk material, which is actually very few. And this type of egg will undergo the first pattern, one of the patterns of the cleavage, which is called the holoplastic cleavage. And what's the meaning of holoplastic cleavage? Plastic means comes from blastomeres, and holo means all. That's mean or total. That's mean the whole cell will be cleaved, not part of it. So the cleavage furrow will extend through the entire egg. And in these type of embryos, since the amount of yolk is very few, so these type of embryos need to find another source or another way of obtaining food. So that's why some of these embryos will have a larval stage. Other embryos, like mammals, for example, will have a placenta to connect the embryo to the mother to get fed, since the amount of yolk is very few. So for the holoplastic cleavage, it's actually can be divided into 4 other cleavage. So understood now that holoplastic means the whole cells will be completely cleaved. So we have radial cleavage, radial holoplastic, we have spiral holoplastic cleavage, we have bilateral holoplastic cleavage, and we have rotational holoplastic cleavage. And actually, these depend actually in the absence of a large concentration of yolk. So because it moves with that there is absence or very few amount of yolk, these 4 types of holoplastic cleavage occur in different types of the organism. And if you in different types of organism, and if you remember, we said one of the parameters that determine the pattern of cleavage is the timing and the location of the spindle fiber formation. Okay. Some other egg type, they have moderate amount of yolk that occur in the vegetal pole. This type is called meso, and meso means moderate amount of lecythal, which is the yolk. And this actually goes as well by the holoplastic cleavage. Eggs of insects, fish, reptiles, and birds, most of the cell are made up of yolk, have good amount of yolk. This type of cleavage and these type of eggs will undergo a type of cleavage which is called meroplastic cleavage. In this meroplastic cleavage, only a portion of the cytoplasm, which actually this small portion which is free from the yolk material, this portion of cytoplasm will be cleaved, and the cleavage furrow will not penetrate into the region which have a yolk. That's why this type of cleavage cannot be said holoplastic cleavage, because the whole cell will not be cleaved. And in this type of embryos, the yolk is very sufficient to nourish this animal until hatch and form the juvenile stage of the adult animal in these animals. For eggs of insects have a peculiar type of egg which is called central lecythal egg, in which the yolk is concentrated in the central region of the cytoplasm. So in this case, the cytoplasm, which will be free from the yolk, will be the marginal region or the cortical region of the egg. At this location, the cleavage furrow occur. So cleavage occur only in the rim of the cytoplasm around the periphery of the cell, which actually the regions which have no yolk. And this type of cleavage will be called superficial cleavage. Then we have the egg of birds and fishes have only one small area of the egg that is free of yolk. And this is called telo. And telo means it came from the word terminal at the end, tarafi. Telolecythal egg. And of course, since this region only is the free region of, it is the free region, it is the yolk-free region at the egg. So this cleavage will undergo a peculiar type of cleavage is called discoidal, because actually the cleavage is occurring on a region that appears like a disc, a discoidal cleavage. بالنسبه للبيردز يا جماعه، طبعا اشهر مثال بالنسبه للبيردز البيضه اللي احنا بناكلها. عارفين البيض اللي احنا بناكله؟ اوكي؟
Yes.
حد لاحظ البيضه لما بيقشرها مش في من فوق حته مبططه كده من فوق بتبقى؟
Pointed يعني؟
لا مبططه فلات كده من فوق شويه. اوكي؟ طيب.
مش محل ما فيه اليوك بيكون يعني قصدك؟
محل فيه اليوك.
يا مظبوط.
اوكي. Because as you said the cleavage occur in the discoidal part، يعني الحته اللي بتبقى فري من يوك، لان معظم البويضه بتبقى ايه؟ يوك، اللي هو الصفار اللي احنا بناكله هو ده اليوك. اوكي. في حد عنده اي لخبطه ما بين ان احنا نربط التايب اوف ايج مع التايب اوف كليفج او الباترن اوف كليفج؟ حد عنده لخبطه في الجزء ده؟
دكتور يس كويستشن. هلا نحن حكينا عنها عم نحكي تايبس، في عنا نحن يا التايب اوف ديستريبيوشن لليوك او الاماونت اوف يوك. مزبوط. مثلا الايزوليسيثل والتلوليسيثل والسنتروليسيثل. مثلا الميزوليسيثل هي خاصه بالاماونت اوف يوك، صح؟ مش بالتايب اوف ديستريبيوشن.
ممتاز صح. لا بس خلي بالك، هم جنرالي انت عندك 4 تايبس. عندك ايزوليسيثال ايج، ميزوليسيثال ايج. ايزو يعني almost few or none، فاهم قصدي؟ ميزو means moderate، تيلو means heavy yolk، فاهم قصدي ولا لا؟
اوكي. سو مثل عم نحكي عن الاماونت اوف يوك.
اماونت،
عم نحكي مثل
فكلهم عباره عن اماونت. اوكي. طيب في حد عنده سؤال تاني؟ حد مش فاهم الفرق ما بين الباترنز اوف كليفج؟ لان احنا هنتكلم عليهم بقى ايه بالدايجرام، نشوف شكلهم دلوقتي. اوكي. نشوف. So let's talk about for the isoleucine egg. As a definition, it's evenly distributed yolk, very few amount of yolk. The first cleavage is the radial cleavage, and this occurs in 2 types of animal, the echinoderms and the amphioxus animal. Okay, so you can see this is the egg, complete cleavage, and this explains why it's called holoplastic. You have 2 cell stage, 4 cell stage, and then 8 cell stage. And this is radial cleavage. Then you have the spiral cleavage. It occurs in annelids, mollusks, and the flatworms. And you need to understand why it's called, you need to be sure. First, this is a holoplastic cleavage as well. Why it's holoplastic cleavage? Because all the cells are divided, not a part of the cell. You can see that zygote have been divided into 2 plasmid, 4 plasmid, 8 plasmid. But why it's called spiral? Because you can see from the radial cleavage, let's have a pointer. You can see from the radial cleavage that when it's a 4 plasmid stage and it is divided transversely to form 8 plasmid stage, that each plasmid is on top of another plasmid, perfectly on top of the bottom plasmid, perfectly on top of the bottom plasmid. The difference here you can see in the spiral cleavage, if these are the 4 plasmid and when it's divided transversely, theoretically it should give 1 plasmid on top and 1 plasmid on bottom as on top. But you can see that no, there is a shifting in position. So when you have 4 plastomeres on top, these plastomeres will rotate. And how we know that these plastomeres, the upper tire of the 4 cells, is rotated? How did we know? Because we can see in the 8 cell stage, these are 8 cell stage because you have 8 plastomeres. You can see that each bottom cells on top, there is the intersection between 2 cells. Each bottom cells, you have the intersection. So normally it should be this cell will be typically on top of this cell. So it's a kind of the top tier of cells have been rotated. فاهمين يا شباب الحته دي؟
Yes.
Because that's why it's called the spiral cleavage. With every successive cleavage at this type of organism, there is a spiral rotation, which is important in the development of these organisms. The third type of cleavage is called bilateral cleavage, and you can understand them. It's actually a type of holoplastic as well, because you can see the general title here. It is the types of holoplastic cleavage, which is mean complete cleavage. So the cells are completely cleaved at every stage. So you can see you have 2 cell stage in the bilateral, which occur in tonicates. You have 2 cell stage, you have then reaching 8 cell stage, and you can see something very peculiar starting from that stage, that the plastomeres start to be aligned as if it is mirror image. So that's why it's called bilateral cleavage. So it gives a similarity, bilateral symmetry in the developing embryo. Okay. The fourth holoplastic cleavage, which is called rotational cleavage, and actually these occur in mammals and nematodes. You can see that actually it's very complicated. So you can see that the pattern actually moves holoplastic until reaching a stage that the cells are rotated to take a certain position and got occupied because they are motile. And actually the figure don't show the rotational behavior of the plastomeres. فاهمين يا جماعه الاربع انواع بتوع الهولوبلاستيك كليفج اللي بيحصلوا في الايزوليسيتال ايج ولا لا؟
Yes, clear.
Okay. Let's talk about the mesolycystal type, the mesolycystal egg, and actually it is a holoplastic as well, because you can see that the plastomeres is actually cleaved. As you can see, normally what's occurring normally, if you have there is no or very few amount of yolk. Normally you have first 2 plasmid, 4 plasmid, and then 8 plasmids. But since there are a moderate vegetal yolk deposition, so you will have a lot of yolk, moderately a lot of yolk at the bottom part of the egg. So this will interfere with the horizontal furrow that occurs in order to convert the 4 plasmid stage into 8 plasmid stage. This interfere will lead to the shifting up of the horizontal cleavage furrow, will lead to a small plasmid on top and larger plasmid in the bottom. And this basically due to the effect of the distribution of the Yuck. So in the mesoskeletal, you can see that all the radial cleavage in amphibians, the frogs, for example, are displaced to the upper less areas of the yolk material. Then we have the meroplastic. Of course, all these previous cleavage patterns are all holoplastic. Then you will go for the meroplastic or the incomplete cleavage. And of course, this occurs because we have a huge amount of yolk. It is the tellular egg. Example is we have one of the example is the cephalopods, mollusks. You have as well bilateral cleavage. There are a dense amount of yolk. So that's why you can see only the cleavage occur on top of the yolk. So you are now looking from the egg from the top view. So you can see a cleavage furrow, and it's kind of like distributing the amount of cytoplasm, and it is not incomplete because you can see an end for the cleavage furrow. It's only partitioning the region which is on top of the egg, of the yolk. You have discoidal cleavage, which is occur in the fish, reptiles, and birds, and you can see only cleavage occur at a region that is on top of the yolk material, which is around it's the disc shape, and you can see that blastomeres, which are formed on top of the yolk, is getting smaller and smaller and smaller because these are actually the only region that undergo cleavage. Then finally, we have the centroplastic cleavage. The central ancestor egg, which actually undergo a meroplastic cleavage type as well, incomplete. If you remember, I've told you that the eggs of insects have the yolk concentrated inside the middle, so you have a superficial cleavage. But the insects have a very peculiar mode of cleavage. They divide the nuclei first. You can see that the nucleus, you have one nucleus in the zygote, and then the nucleus start to divide. You have a lot of nucleus. Then the nucleus will be arranged at the periphery of the yolk material, and then the cytokinesis occur, and this nucleus will be partitioned, and every nucleus will occupy a region of the cytoplasm by cell membranes. فهمنا يا جماعه الاربع انواع دول ولا لا؟
Yes.
يبقى دلوقتي اهم حاجه هتلاحظوا في الانسكتس ايج ان النيوكليوس there are syncytium، او النيوكليوس الاول بيحصل لها ديفيجن، ودي حاجه مختلفه تماما عن بقيه الكليفج باترن. ان النيوكليوس بيحصل لها ديفيجن الاول، وبعدين النيوكليوس دي ترتب نفسها على الاطراف البويضه او اطراف البلاستيلا، وبعدين يبدا هذه النيوكلياي تبدا تتقسم ويظهر سيل ممبرين ما بين النيوكلياي. فهمتوها دي ولا لا؟
Yes.
اوكي. وطبعا ليه الكلام ده بيحصل؟ لان اليوك مركز فين؟ في المنتصف، center of the egg. That's why this type of egg is called centrolysis. Okay, got it? Okay. So here I'm just it's a repetition, but here I'm just showing all type of cleavage in one slide. Okay, ده اللي احنا شرحناه بالظبط، بس انا مجمعهم كلهم عشان الصوره تبقى واضحه ليه كلهم التايبس كلها. And this conclude our lecture for today. I'm waiting for your questions. حد عنده أي سؤال يا شباب بالنسبة للمحاضرة بتاعت النهارده؟
دكتور ما عندي سؤال على اللكتشر، بس كرمال الإكزام
طب لحظة،
كنا عم نحكي.
just hold on.